Hello and welcome to Bio40 Pre Lecture 16. Today we're going to discuss the one gene, one enzyme hypothesis. This is the central dogma of molecular biology. It was coined by Francis Crick in 1956. The central dogma says that information flows from DNA to the messenger molecule RNA to proteins, which have key enzymatic and structural functions. We're going to spend the next few weeks going through this process. First, how is it established that genes contain the information to make enzymes? The idea was first suggested in the early 1900s by Archibald Garrod, who was studying human metabolic diseases. But strong evidence was not provided until the 1940s by George Beadle and Edward Tatum. Garrod studied the rare autosomal recessive disease alkaptonuria. This condition involves a breakdown of cartilage, leading to osteoarthritis, and has the odd effect of causing urine to turn brown or black. The root of these problems is a buildup of the substance homogentisic acid, abbreviated HA. HA is formed as part of the process of getting rid of excess phenylalanine and tyrosine. But HA is normally only around very briefly before being converted to another compound and eventually broken down to just carbon dioxide and water. Garrod hypothesized that affected individuals were lacking the enzyme HA oxidase, needed to continue down this pathway. As a result, HA builds up and causes damage to the affected individual's cartilage. That the disease can be inherited suggests a gene mutation can affect whether or not an enzyme gets made. Beadle and Tatum built on this idea for their one gene, one enzyme hypothesis, wherein each gene has the information for a single enzyme. They set out to support this idea by showing the correlation between genetics and biochemistry. They used a pathway with known intermediates like the one shown below, wherein the amino acid methionine is synthesized from homoserine. Organisms with a mutation in such a pathway would be methionine auxotrophs. If Beadle and Tatum were correct, they should be able to mutate different genes and get rid of different enzymes that catalyze each reaction in the pathway. A missing enzyme can be demonstrated by the buildup of an intermediate, and such mutants could be rescued by adding a different intermediate. For example, a MET2 mutant would not have the enzyme cystathionase 2, which means the intermediate cystathionine would build up. But the addition of later intermediate homocysteine would rescue the mutant because the pathway isn't damaged after that point. Beadle and Tatum chose to mold Neurospora crassa as their model organism, shown here with some cool fluorescent microscopy. Similar to yeast, Neurospora has haploid and diploid stages. Haploid cells exist as either A or alpha mating types. They can reproduce asexually and form these branching structures called mycelia. The mycelia can release spores called conidia that can grow into new mycelia. When an A and alpha cell meet, their nuclei can join to form a diploid cell. The diploid cell then undergoes meiosis to yield 4 new haploid cells. These undergo one more round of replication and division for a total of 8 haploid cells called ascospores, contained in a structure called a fruiting body. These ascospores are released to form new mycelia. Beadle and Tatum took wild-type Neurospora conidia and mutagenized them through exposure to X-rays. Then they mated them with wild-type cells of the opposite mating type and allowed fruiting bodies to form. They dissected the fruiting bodies and moved each ascospore one by one into different tubes with complete or rich media. Both auxotrophs and prototrophs will grow on complete media, so this step is just to give a chance for the ascospores to grow and divide. Next, a few cells from each sample were transferred to minimal media, where wild-type cells would grow fine, but nutritional auxotrophs would not. These auxotrophs could be deficient in making amino acids, nitrogenous bases, vitamins, etc. So the next step was to attempt to grow the mutants on minimal media with one of these components added in. Beadle and Tatum were particularly interested in amino acid auxotrophs, and looked for mutants that could be rescued by supplementing the media with said amino acids. Then, to figure out which specific pathway was damaged, they moved the mutants to 20 different tubes, each supplemented with just one of the amino acids. In this case, you can see growth only occurs in the tube with tryptophan added, so the mutation is in the gene necessary for tryptophan synthesis. In this way, Beadle and Tatum were able to get a bunch of mutants for each amino acid synthesis pathway. By now, I'm sure you can tell how they figured out which mutants were in the same gene and which were in different genes. Let's look at the example of methionine auxotrophs. They identified mutants with mutations in 4 different genes, termed MET2, MET3, MET5, and MET8. Because the biochemistry of the methionine synthesis pathway was well known, They could show which gene corresponded to which step in the pathway using the process of intermediate rescue. Of course, any methionine auxotroph could grow when methionine is added to the media, but really, the mutants could be rescued with the addition of any intermediate that appears in the pathway after the missing enzyme. This table shows the growth response of the different mutants when each intermediate is added. A plus sign means growth is possible, but a minus means no growth. MET5 mutants are rescued by any intermediate. So MET5 must encode the enzyme that catalyzes the earliest step in the pathway, converting homoserine to O-acetylhomoserine. O-acetylhomoserine doesn't rescue MET3, so the enzyme MET3 encodes must act after it in the pathway. But all the other intermediates rescue MET3 mutants, so this enzyme catalyzes the next step. Likewise, we can conclude that MET2 comes next, and MET8, which is only rescued by methionine, must catalyze the last step in the pathway. We'll discuss how to use these intermediate rescue tables more in class. Such data should also be backed up by precursor accumulation. If MET5 is mutated, homoserine will accumulate. If MET2 is mutated, cystathione will accumulate, and so on. This is all strong evidence that each gene is responsible for making one enzyme in the pathway. But over the years, as we've learned more, the situation has grown more complicated than that. First of all, not all proteins are enzymes that catalyze reactions. Some play structural roles, or do mechanical work, or have other roles. So it might be a little more accurate to say one gene, one protein. Also, a lot of proteins don't function alone. Many act in groups, called complexes. E. coli's DNA polymerase III, which we've discussed, is actually composed of 13 different polypeptide chains encoded by 8 different genes. So maybe we can say one gene, one polypeptide. But then there are some genes that can actually make multiple different polypeptides using a process called alternative splicing. More on that in a future lecture. Plus there are redundant genes, which means that there are multiple genes that encode proteins that can carry out the same enzymatic function. It's particularly hard to screen for such genes because mutating just one of them doesn't alter the phenotype. Finally, not all genes even encode proteins. Some functions are carried out by RNA molecules that are never translated into proteins. We'll talk about many of these non-coding RNAs later in the course. Now, except for that last case, which only accounts for a small portion of genes, genes contain the information to make proteins. Proteins are linear polymers of amino acids linked by peptide bonds, also called polypeptides. Specifically, genes direct the identity and order of the amino acids within a polypeptide chain. There are 20 different amino acids that form the building blocks of proteins. Each amino acid consists of a basic amino group and an acidic carboxyl group. Joining these two groups is the alpha carbon, which is also attached to a hydrogen atom, and an R group, which is also called a side chain. The R group is what differs between the 20 amino acids. Amino acids are joined together by peptide bonds. The carboxyl carbon of one amino acid becomes covalently bound to the amino nitrogen of the next amino acid. In the process, a water molecule is released, so this is sometimes called a dehydration or condensation reaction. The amino acids within the chain are then referred to as residues. Once again, we are dealing with a polymer that has polarity. The end of the polypeptide with a free amino group is called the amino or N-terminus. The other end, with the free carboxyl, is a carboxyl or C-terminus. Now let's take a look at the different R groups that exist. 3 are basic or positively charged: lysine, arginine, and histidine. Don't worry about memorizing the specific side chains yet. That's for next semester. 2 R groups are acidic or negatively charged: aspartic acid and glutamic acid, also called aspartate and glutamate. You can see the structures are very similar, differing only by a single carbon in the length of the side chain. This is what I meant a few lectures ago when I said there can be neutral mutations where switching amino acids probably won't have an effect. A lot of the time, you can substitute an aspartate for a glutamate or vice versa without consequence. Though this isn't the case 100% of the time, it's certainly less likely to cause a problem than if you were to substitute a negatively charged amino acid with a positively charged one. For the uncharged side chains, we separate them into polar and nonpolar. These are the 6 uncharged polar amino acids: tyrosine, serine, threonine, asparagine, glutamine, and cysteine. Again, amino acid substitutions within a group are more likely to be tolerated, though swapping a small side chain with a large one, or vice versa, certainly could cause some damage. Later on, you'll also learn about certain cases where one specific amino acid is required at a given position. The remaining 9 amino acids are uncharged nonpolar: tryptophan, phenylalanine, glycine, alanine, valine, isoleucine, leucine, methionine, and proline. The same general guidelines about substitutions within a group apply here too. Finally, despite one being polar and the other 2 nonpolar, the 3 amino acids with phenyl groups, tyrosine, tryptophan, and phenylalanine, are often grouped together as the aromatic amino acids. As we've gone through, you've probably noticed that each amino acid has a distinct 1-letter and 3-letter abbreviation. But you also don't have to worry about memorizing those. Yet. The sequence of amino acids within the polypeptide chain is called the primary structure. But the 3-dimensional protein structure is much more complicated than that for DNA. Unlike DNA, proteins can have many different structures depending on the sequence of amino acids. In fact, how to predict a protein's 3-dimensional structure based on the amino acid sequence is one of the great problems being worked on today. Secondary structure looks at local spatial relationships within a polypeptide. The 2 main types of secondary structure are alpha helices and beta sheets. Both are the result of hydrogen bonding within the polypeptide chain. Alpha helices involve hydrogen bonding between the NH group of one amino acid and the CO group of the amino acid 4 positions earlier in the chain. Beta-pleated sheets also involve hydrogen bonding between NH and CO groups, but here the bonds connect adjacent sheets of amino acids. These structures were first proposed by Linus Pauling, and were part of the work that earned him a Nobel Prize. Next there is tertiary structure, which is a polypeptide's final 3-dimensional structure. This is the level that is most difficult to predict from the primary sequence. Much of the X-ray crystallography work performed these days is done to find out this information. Such studies are so important because a protein's structure can reveal a great deal about how it functions. Finally, there is also quaternary structure. This is the 3-dimensional structure of protein complexes consisting of multiple polypeptide chains. I want to end this lecture by addressing the polarities we've seen so far. We've already talked about DNA being synthesized 5' to 3', and soon we'll see the same is true when RNA is copied from DNA. Next time, we're going to start discussing how the DNA/RNA sequence is interpreted as an amino acid sequence. The messenger RNA, or mRNA, is read 5' to 3', and the polypeptide chain is synthesized from the N-terminus to the C-terminus. This tells us that the 5' end of the DNA sequence corresponds to the N-terminus of the protein it codes for. Okay, that's it for now. Next time, we finally learn how exactly a sequence of DNA bases can be interpreted as a series of amino acids. We look at the genetic code.